Let us take you through a day in the laboratory in the city of palaces, where the hospital never sleeps and deep within, the laboratory carries on its quiet, constant work. Through the night, samples arrive, instruments run, and dedicated teams work behind the scenes, ensuring that every result reaches the clinician in time.
As the first light touches the city, Mysore begins to awaken.
The hospital slowly fills with life, the patients arrive, the corridors grow busy, and the consultants begin their day. And at the heart of it all, the laboratory continues its work, turning samples into answers that guide care.
Before any result reaches a patient, it first goes through our stringent internal quality control process. We run QC 3 times a day, and patient testing begins only after the QC is within acceptable limits. We follow a proactive, structured quality assurance program that is in accordance with NABL guidelines.
By participating in monthly e-cards, we are able to appreciate external accountability and benchmarking. This kind of training shapes our mind that quality isn't an option but defines our professional integrity.
In our laboratory, the training is a continuous and structured process. Through this process, we are not just running the tests in our lab. We are creating very able, competent and also ethical laboratory professionals.
We take pride in providing skill-based training in the laboratory for undergraduates, postgraduates across the disciplines in par with international standards.
Just now we have received DBS card sample for newborn screening. Newborn screening starts in NICU, where the sample will be collected by heat-treat method under strict precautions. After proper drying, this DBS sample will be transported to our laboratory.
In the lab, we punch a sample, perform elution and initially screen 7 essential metabolic parameters using ELISA-based assay for early detection of treatable disorders.
For extended newborn screening, we employ LCV tandem mass spectrometry platform that enables the simultaneous detection of various amino acids, organic acids and other fatty acid oxidation disorders. Currently, our LC-MS platform is processing the samples for therapeutic drug monitoring.
At this hour, we are receiving high volume of OPD samples from multi-speciality across the hospitals like urology, nephrology, gastroenterology and more. All our routine biochemistry parameters are processed through fully automated biochemistry analysis.
Critical values are immediately communicated. The validate reports are delivered systematically to hospitals, concerns and departments.
Now we have received samples from the oncology OPD for the tumor marker analysis. After the QC validation and barcode verification, we are going to load this sample into the immunoassay analyzers.
We receive the ABG sample in a heparinized syringe. It is transported in the eye slurry to prevent the gas integrity and to preserve the metabolic changes. It is immediately locked, gently mixed and loaded into the blood gas analyzer without any delay.
Today we received 5 samples for serum protein electrophoresis. This helps us to identify abnormal protein fractions. Whenever required, we proceed with immunofixation electrophoresis to characterize specific immunoglobulin type.
While interpreting the HbA1c reports, we go through each of the graph. Today we have received about 4 samples for HbA2. While interpreting the HbA2 values, we look at the retention time, the graphs, we compare this with the red cell indices, and whenever we are in doubt, we discuss this with the clinician.
Also, our osmometer measures serum and urine osmolarity with high precision, which helps to assess fluid imbalance, electrolyte imbalance and critical care status within minutes.
In our genome facility, a wide spectrum of assays are performed like karyotyping, RT-PCR, microarray, whole exome sequencing, Sanger sequencing as and when the samples are received. Our wet lab team follows stringent SOPs for nucleic acid extraction, quantification, library preparation, amplification and sequencing.
Once sequencing or molecular analysis is completed, so the data moves to our bioinformatics and molecular genetics team for tertiary analysis.
We have just received a start sample from the casualty for a suspected myocardial infarction. After immediate barcode verification, the sample is prioritized and loaded into the chemistry analyzer under emergency protocol.
Hello casualty, we are from the Department of Biochemistry. The start sample for TROP-T with the request number of 422570. The value is high, kindly check the LIS.
Our laboratory generates a rich repository of clinically relevant data every day. When ethically integrated and analyzed, this data supports translational research, strengthens academic output, and contributes to significant publications that ultimately improve healthcare delivery.
From midnight to midnight, our laboratory functions with unwavering vigilance and efficiency, supporting every ward, every emergency, and every patient who depends on timely diagnosis. At the Department of Biochemistry, science meets compassion, data becomes diagnosis, and precision becomes patient care.